So when we're talking about agglutination, we're talking about antibodies which have already been developed. Okay, they've already been developed. Okay, they are ready to go. They are matching with the antigen. Okay, that's what we're talking about. The antibody doesn't need to change its tertiary structure or do anything like that. Phagocytosis is going to occur. So over here, this is already, so let's say you've been vaccinated, you've already got the antibodies built in within your body. If you've got the antibodies already built in within your body, they're going to be able to attack the antigens of the sort of the pathogens which come in very much easily. If it's your first time being exposed to this sort of pathogen, your antibodies aren't going to be ready. So this response won't occur straight away. Your body will have to go through your cell-mediated and humoral response in order for the antibodies to be produced first. Then once the antibodies have been produced, they can go and agglutinate, they can go and attack the leftover pathogens um, and sort of get rid of them by phagocytosis. Phagocytosis will engulf them, and then cytotoxic T cells will come and get rid of them. Okay, so it's not specifically, let's say if you've got, let's say COVID, let's say you have COVID, and let's say you've not been vaccinated against COVID. This process won't occur straight away. You will start feeling a bit ill first because your antibodies aren't ready within your body. And then what's going to happen is that your cell-mediated response occurs, your humoral response occurs, then your antibodies are ready. Then what they will do is they will bind to the antigens and they will get rid of those antigens. Agglutination occurs then. It doesn't really occur at the start. It occurs then, and then it locates phagocytes and cytotoxic T cells to kill the pathogen. Okay, I'm just going to give anyone else who's having a read of this question and having a go at it 30 seconds, and then I'm going to start going through the answers. Okay, so I'm going to go through the answers now. Okay, so it says there are 4 main blood groups in the human ABO blood group system. The table below shows the basis on which each of these blood groups is classified. So we have the ABO blood group. This sort of links in with another topic that you will study about blood groups AB, or O. Okay, and then the antigens which are present on the red blood cells. When you have a blood group of A, the antigen present is A. When you have a blood group of B, the antigens present are B. And then when you have AB, the antigens present are A and B. And then O, you don't have A or B antigens present. The antibodies which are present in your blood plasma when you have a blood group of A is anti-B. Okay, so they're making sure that you don't have any sort of B antigens present within your blood. Okay, so with B you have anti-A, with AB you have anti-A, you don't have anti-A or anti-B, and then O you have anti-A and anti-B. Now it says a transfusion of blood from a blood group A donor to a blood group B recipient would cause agglutination of the donated red blood cells. Use information in the table above to explain why. So let's think about this question. Now, we've got a blood group B recipient. Now, when we have a blood group of B, over here we've got a blood group of B, we know that the antigens that would be present in the patient's blood would be B antigens, and they would have anti-A antibodies present within their blood. Now, when we're getting blood from blood group A, we know a person with a blood group of A has A antigens in the red blood cells and anti-B antibodies. Now, when we're taking A and we're putting it into B, we're putting A antigens within blood that has anti-A antibodies. Okay, because we're putting A antigens from blood group A into blood group B recipient. Blood group B has anti-A antibodies, which means that it will go attack A antigens. Okay, so what's going to happen is that when this blood transfusion occurs, is that the anti-A antibodies, they are going to bind with the A antigens and a complex is going to form. And once one complex is formed, the cells are going to agglutinate. Okay, they're going to clump together, they're going to join to each other, and agglutination is going to happen because you have foreign antigens present within the blood of the recipient. Okay, you have A antigens present. So antibodies are going to come in and they're going to agglutinate. Multiple antibodies are going to join to that same A antigens and they're going to clump together so phagocytosis can occur and cytotoxic T cells can get activated. So what's going to happen over here is that because we're putting the blood into blood group B recipient, the blood group B recipient has anti-A antibodies. Okay, and we're putting antigen A into the recipient's blood. So what's going to happen is that the anti-A antibodies from recipient will bind to antigen A from your donor. Okay, that's what's going to happen. So the antibodies are binding to the antigens. This is going to cause agglutination. Okay, it's going to cause the complex to agglutinate because you've got foreign antigens. The antibodies are going to clump together. Okay, so what's going to happen? This because we're talking about red blood cells here, we would say that this causes the red blood cells to clump together and therefore agglutination occurs. Okay, so that's what's going to happen. If I show you the mock scheme for this question, it says binding between antigen A and anti-A. So because we're putting blood group A into blood group B recipient, blood group A has A antigens, and that's going to bind with the anti-A antibodies that blood group B has. And when they bind, that's basically what this first point is saying. And once they've bound to each other, that's going to trigger an immune response, causing the cells to clump together. So agglutination and this key word of clump go hand in hand with each other. Okay, so that's where your second mark is coming from. Okay, because once an immune response has been triggered, agglutination happens, which means that the normal sort of red blood cells, they will clump together. They will clump together, causing phagocytes to be recruited. Okay, so moving on to our next question. So this isn't sort of directly part of this topic, but the context of it is related to immunity. So this one is a 3-marker question. Again, I want you guys to have a read of it first and attempt it as well. And then I'm going to start going through the answers in a couple of minutes. If you want to type your answer into the Q&A, feel free to do so. Okay, I'm going to start going through the answers now. Okay, if we go back, it says ignore reference to anti-B in donor. So what's going to happen, because you're putting in anti from blood group A is going to be transported into a blood group B recipient. That blood group A contains the blood group A contains anti-B antibodies present. Now the donor has blood group A, the recipient has blood group B. So we're talking about the effect that it will have on the recipient. Okay, now the recipient had, well, over here the only sort of decisive sort of answer that you can give is because antigen A is being transported into patient donor the recipient's blood. It's going to be triggered by anti-A antibodies. Antibody B is also being transported into the blood, but the recipient has sort of B antigens. So it's not allowing a direct reference, because you can't make that conclusion that anti-B antibodies will definitely trigger an immune response in the recipient. We can only make the conclusion from the information that has been given that if you transport antigen A into the recipient, and that recipient specifically has anti-A antibodies, that's going to cause a direct impact, direct relationship between the antigen A, which has been transported within your body. We can't make a decisive conclusion that because anti-B antibodies has also been transported into your body, that's going to affect the B antigens that are also present in the body. Okay, that's why it's saying ignore reference to anti-B in your donor, because we can't make that conclusion decisively based on the results that we have been given in the table. We can only make the link between A and anti-A. Okay, so if you have a look at this question over here, it says some hospital patients suffer from diarrhea caused by infection with the bacterium. I'm not even going to try spell that word out. The bacteria releases toxins. These toxins causes diarrhea. The toxins damage the cells lining the ileum, causing them to lose their microvilli. The damage to the cells reduces the absorption of the products of digestion and reduces the absorption of water, resulting in diarrhea. Explain why the damage to the cells in the ileum reduces absorption of the products of digestion and why this reduces the absorption of water. So they like putting in these questions, especially worded questions, because sometimes you end up reading them and you end up sort of being lost within the question. The key aspect is looking at the key phrases, key wordings, key things that you might have come across before and focusing on them. So definitely microvilli you have probably come across before. Okay, microvilli you would have come across at GCSE as well. Okay, it says the toxins damage the cells lining the ileum, causing them to lose their microvilli. Now, if we're talking about absorption and we've damaged the microvilli, obviously the purpose of microvilli in our cells is to increase the surface area. So the first point you're going to straight away put down is that because there have been damage to your microvilli, that's going to reduce the surface area. Okay, so due to damage of the microvilli, the surface area is going to be reduced. Okay, that's the first decisive conclusion which you can come up with straight away. Now, if the surface area has been reduced, let's read on. It says the damage to the cells reduces the absorption of the products of digestion and reduces the absorption of water. So what's happening is that in our ileum, the microvilli has been damaged. That's reduced the surface area. The purpose of your microvilli is to have a large surface area so you can absorb your water, you can absorb your products of digestion. We've reduced the surface area. This means that water can't move into the ileum. Okay, water can no longer move into the ileum, which means that the water potential of the ileum has been reduced. Okay, so we would say that the water potential of ileum has been reduced. Okay, so the water potential of the ileum has been reduced. Now, if the water potential in the ileum being reduced, what's going to happen? So remember, this is happening within your small intestine. The water potential of the ileum has been reduced. That means that the water potential outside the ileum is going to be much higher, which means that the water will move out of the ileum by osmosis, which means that more water will be excreted. Okay, so this means water will move out of the ileum. And remember, whenever you're talking about movement of water, always say osmosis. Osmosis is normally always underlined in mock schemes. So this means water will move out of the ileum by osmosis, which means that more will be excreted, resulting in diarrhea. More water will be excreted, resulting in diarrhea. Okay, so that's what you would have to say there. If I show you the mark scheme for this question, so the first mark is coming from saying that it's a reduced surface area, because we specifically have to spot that microvilli statement, and we have to think to ourselves, we need to know about microvilli that has been damaged. So what? If that's been damaged, that's going to reduce your surface area, because that's the purpose why we have microvilli. Now, if the microvilli has been damaged and the surface area has been reduced, less water can move into the ileum itself. Now, the ileum plays a really important role in making sure that you don't have diarrhea. It has microvilli to increase the surface area for that specific reason too. So enough water can be absorbed into the ileum, so not a lot of water can be excreted out. That's your purpose of your ileum. If the surface area has been reduced because the microvilli has been damaged, then obviously the water potential um, inside the ileum is going to decrease. Okay, the water potential inside the ileum is going to decrease, and what's going to happen is that water is going to move out into the ileum by osmosis. Okay, I've just realized I've made a slight error over here. Move out of into the ileum by osmosis. So then more water is excreted, resulting in diarrhea. Okay, so we've got decrease in your water potential because we've damaged the microvilli, which means that we've reduced the surface area. Now, because now the ileum, that's basically the ileum is the terminal part of your small intestine. Your small intestine has different parts. Your ending part is your ileum, and basically your ileum will then connect up to your large intestine, and it goes through that tract. What happens if not enough water moves out of the ileum, then a lot of water will stay in, which means that a lot of water will move into your large intestine, which means a lot of water will get excreted. So because we've reduced the water potential of the water in the ileum, that means more water will move into the ileum. If more water moves into the ileum, that means more will go into the large intestine, which means more water will be excreted. So your third mark is coming from saying that water moves into the ileum by osmosis. Okay, so those are where your 3 marks are coming from. Okay, So moving on to our next question. So as I said, you could be given an image of an antibody. Over here, we are given an image of an antibody. It says the diagram below shows the structure of an antibody. It says label the diagram above with an X to show where the antigen antibody complex forms. Remember, the antigen antibody complex forms at the variable region. Okay, so what you would say is you just have to draw an X where it forms. It forms at this region over here, at the ends of the antibody. That's where the variable regions are. So you would just write an X at both ends of the Y shape. Okay, and there's an important label that we've not actually been through over here, and that's the disulfide bridge. Okay, and the reason I'm not going through, I've picked out a specific exam question that's asking you the purpose of that disulfide bridge. Okay, and this is the function of the disulfide bridge. We're going to look at the next exam question and we'll finish on that note. So it says in this diagram over here, we've got a disulfide bridge labeled. As you can see, we've got this disulfide bridge, which is being labeled. What is the purpose of that disulfide bridge? Now, as you can see, we've got a disulfide bridge over here and over here as well. These are disulfide bridges. Now, the purpose of this disulfide bridges, as you can see, it's connecting. We know we have one polypeptide chain, two polypeptide chain, three polypeptide chain, and then four polypeptide chain. We have four polypeptide chains, which makes this a quaternary structure antibody. Now, all of them have to be connected. Okay, they can't just be floating around, they have to be connected to each other. And the way that they are connected to each other is that they have this disulfide bridge. The disulfide bridge is what connects the different polypeptide chains together. It joins the different polypeptide chains together. So over here, it says a disulfide bridge is located in the diagram above. What is the role of the disulfide bridge in forming the quaternary structure of an antibody? The reason is the disulfide bridges are the ones which join the two polypeptides together. They join different polypeptides together. You don't want the four polypeptide chains hanging loosely to each other, sort of floating around. You want them to be in one structure, joined together. And that's what the disulfide bridges are there for. They are there to join the four different polypeptide chains together.